Hi, good afternoon, class. I'll be recording on the chapter 3, and this chapter is called as DNA, the blueprint of life, from genes to proteins. So genes would mean a segment of DNA that has capability to code uh, proteins. Not all DNA codes for proteins. Uh, there are only selective. So let's say if we have 46 chromosomes, chromosomes are made up of DNA and proteins. So not 100% of the DNA that is present in the chromosome codes for protein. Only selective DNA codes for proteins. So let's start this chapter, but before I start this chapter, I would like you to just review the syllabus, at what point we are. So we finished, until now, we finished chapters 1 and 2, which were on biomolecules and cellular structure and cell division. And now we are going into chapter 3. So today, my plan is to record for 3 chapters. Number 1, that's chapter 3, DNA, the blueprint of life. Then patterns of inheritance and molecular cloning technique. So you'll get 3 separate recordings, and that will be the one of the few of the learning activities for next week. Okay, so let's get started. So before I start any chapter, I like to give a little bit of concept. So remember at what point we started this class. We started this class or this course by starting from learning about atoms, molecules, how they're chemically bonded, and we also looked at different types of chemical bonding. And then we went into biomolecules, particularly protein, lipids, carbs, and nucleic acid. And then after that, we went into looking into cell structure of bacterial cells and plant cells and human cells. And then we looked at the function of various organelles. And after that, we entered into cell division, starting from DNA replication, and then mitosis, and then meiosis. And then we did a detailed review. I hope you got a chance to review the assignments, my recording I meant. So these we have covered until now. Now, based upon this knowledge of these topics, we go into the next chapter, which is third chapter, which is DNA. Print of life. Now, why DNA is referred as blueprint of life? Because DNA has all the information. DNA has all the information coded that when it unfolds in the journey of life of any organism, whether it's bacteria, plants, or humans, or animals, DNA has those mysterious programming that helps to in the development or division or cell growth. So DNA is the information center, hub for all the knowledge that would unfold during the course of time. And what is a blueprint? Blueprint means whenever a big project is coming up, let's say a building, a very big, ambitious project, instead of directly landing on a project from day 0, they would have to do the planning, right? Because planning would lead to budgeting and how much investment, so on and so forth. So, and they would like to build a model structure, because if you spend a lot of resources and the model is not clear, and you say that you'll take one day at a time, then it could be a successful project, but it has higher chances of failure. So when you build this planning, budgeting, resources, human support, you model the small structure, you are in good shape to succeed. So that is called blueprint before you really start the project. Everything is every bits and pieces are very clear, and it could be just making a model as you go on. So DNA serves as a blueprint of our life. It has all the information. For example, in some diseases, if so DNA has, just like English literature, which has 26 alphabet, DNA has only 4 alphabets, A, T, G, C. These 4, what are these? These are nitrogenous bases that exist in the DNA as nucleotides. Ultimately, they become a language. These 4 nucleotides or nitrogenous bases, they become a language, and mainly for proteins. So, and these proteins do a huge work for our body. Proteins are very important. So, coming back, the chapter is Blueprint of Life, DNA. And also, in another way, you can say that this chapter focuses on a journey from genes to proteins. How genes, and genes would mean how DNA would serve as a template for the synthesis of proteins. This was the background. Now, let's go to the next. So DNA, deoxyribonucleic acid, it has 2 functions. Number 1, the first function is DNA replication. Okay? And DNA replication would mean when DNA serves as a template for the synthesis of new DNA, daughter DNA. That's before when the cell divides. Now, this process is carried out by an enzyme called as DNA polymerase. Right? So a DNA is duplicated into DNA by an enzyme called as DNA polymerase. The second function of DNA is transcription. And transcription is a process where DNA serves as a template for the generation of RNA. And like replication, transcription is also an enzymatic process, and this is carried out by an enzyme called as RNA polymerase. So you can clearly see the difference here. DNA is the starting point. DNA is the starting point, but it has 2 responsibilities. One, it has to be replicated, and second, it has to serve as a template for transcription. Now, why transcription? It's because transcription would serve as a RNA would serve as a template for translation. And what is translation? Translation is a process where RNA serves as a template for the synthesis of proteins. So RNA serves as a template for the synthesis of proteins. This chapter will mostly focus on this part. What are those parts? First is transcription, and the second is translation. So we are covering two steps in this chapter. This is a small chapter, but it's a very important chapter because if this doesn't exist, I believe if this process, this pathway, both replication and transcription and translation, if they don't exist, I don't know how a cell, a bacteria would come into, how a living things would anyway germinate on this planet. So these are very important steps. This is the topic of intense research for developing new medicines. And now let's correlate to today's pandemic. That so today's pandemic, we are dealing with COVID-19, and as of today, there are many deaths that are happening across the globe. So unless we understand the biology of COVID-19, we can't develop the vaccines. And so COVID-19 has a genetic material as RNA, and since it is RNA, they have a different enzyme for the growth of RNA, for the multiplication of RNA. They have an enzyme called as RNA dependent RNA polymerase. So inside the host, when this virus has to replicate, has to divide, they have to use this enzyme, RNA dependent RNA polymerase. So so with this basic information, I want to spend little time with respect to difference between transcription, how transcription occurs in eukaryotes, how transcription occurs in prokaryote, that is bacteria. So bacteria, we know that there are no, in bacteria there is no nucleus. So let's say we are talking about transcription in bacteria and in human cells, that would represent a prokaryote. So since bacteria, they don't have nucleus, so transcription and translation both. So I will write transcription as this way and translation as like this, translation. So both transcription and translation in bacteria, it happens in cytosol or cytoplasm. Whereas in humans, transcription occurs in nucleus. Transcription occurs in nucleus because the DNA. Why? Because the DNA is present in the nucleus. And translation in bacteria as well as so translation in bacteria as well as in human cells or in cytosol. So translation is common. Now the question comes, why translation takes place in cytoplasm? Remember, I mentioned to you that one of the major organelles that is responsible for protein synthesis is called as ribosome. Now this ribosome is present in cytoplasm. This ribosome is present in cytoplasm. In cytoplasm. So let's say in human cells, if the DNA is present in the nucleus, the DNA has to travel from the nucleus. The DNA has to travel from the nucleus to the ribosome, right? Now DNA cannot travel, so DNA has to transcribe. And what would transcribe means? That DNA would serve as a template to generate another molecule called as RNA, right? And this RNA would travel from nucleus to the ribosome for getting translated. So because translation, protein synthesis synthesis occurs on ribosome. Ribosome becomes the major site for translation. So this is the difference between bacterial and eukaryotes in terms of transcription. Now, we all know by now that DNA is made up of 4 bases, and RNA is made up of also 4 bases, but there's a major difference, and the major difference is that in DNA it is ATGC, whereas in RNA it is AUGC. So this uracil, the flip between uracil and thymine is a major consequential thing that happens in transcription. So what is the logic for transcription? The logic for transcription is that, let's say if there is a DNA which is made up of 2 strands, and we have bases like AT, GC. GC has triple bases, triple bond. So GC has a triple bond. So A adenine is connected to thymine by double bond, and G guanine is connected to cytosine by triple bond, triple hydrogen bond. 1 hydrogen bond, 2nd hydrogen bond, and 3rd hydrogen bond between G and C. Now, these can, so let's say if these serves as a so let's consider this as DNA. So when these 2 strands could be indicated as A and B. So let's say if the strand B is getting transcribed or undergoing transcription, then the T will code for A and C will code for G, right? And let's say if here it was A, then this in the RNA, this will code for uracil. So it is a complementary. Complementary means, complementary is written as complementary. Logic is that if the DNA has adenine, then the corresponding nucleotide on the RNA would be uracil. If the DNA has T, the corresponding nucleotide on RNA would be A. If the DNA has C, then the corresponding will have G, so on and so forth. So this relationship between the DNA and the new RNA is called as complementarity, and that's the basis for transcription. So number 1, in human cells, transcription occurs in the nucleus. Transcription is mediated by an enzyme called as RNA polymerase. And the logic for transcription is that if the DNA strand has a nucleotide adenine, the corresponding RNA strand would be uracil. If it has T, it has A. So that's the way transcription occurs. And I will also post YouTube videos. So you can correlate my lecture with the YouTube videos, which would have a better animation than at this point what I have. So now this is this process of translation or protein synthesis is a teamwork. Teamwork, what I mean, that DNA serves as a template to synthesize 3 kinds of RNA. So until now, I was using the word RNA. Just But actually there are three types of RNAs that mediates the process of translation. The first type of RNA is referred as mRNA. mRNA. The second kind of RNA is referred as tRNA. And the third kind of RNA is called as rRNA. Right? Now, as the name indicates, M stands for messenger RNA. E stands for transfer RNA. And R stands for ribosomal RNA. So one piece of DNA serves as a template for synthesizing three different kinds of RNA: mRNA, tRNA, and rRNA. Messenger, transfer, ribosomal RNA. So now, what is the specific role of these RNAs? So let's say mRNA. mRNA serves as a template for amino acids. mRNA serves as a template for amino acids sequence. Every protein has a sequence of amino acid. So, and this sequence of amino acid that is present in a protein is dictated by mRNA. And mRNA is what? Sequence of ribonucleotide, ATU, AUGC. It doesn't have, RNA don't have T. So AUGC and permutation and combination of that. I'll come back to that later. The second kind of RNA is called as tRNA. It looks like this, tRNA. The function of tRNA, or rather transfer RNA, is to carry amino acids from cytosol to the surface of ribosomes. So let's say if there is a cell. So let's say if there is a cell, and this cell has a ribosome, right? So on this ribosome, mRNA will come from the nucleus, remember? So there is a nucleus. This nucleus has a DNA, double helical DNA. This will undergo transcription with the help of RNA polymerase, and then these RNAs, mRNA, tRNA, they all will come to cytoplasm, and from the cytoplasm, they'll work together and they'll go to the ribosome. Right? So mRNA, this is mRNA that will serve as a template for the sequence of amino acids, and tRNA will bring amino acids. So in a cell, there are in the cytoplasm there are there are 20 types of amino acids which are present in the cell. 20 different types. Now, who will bring them to the ribosome? So the tRNA, the transfer RNA, it works just like UPS or USPS. It will take the package and deliver to a specific address. So what is the package? The package is respective amino acids. And where it will deliver to? It will deliver to the to the ribosome. So you have the mRNA, you have the amino acids now coming to the ribosome. Now, what is the role of rRNA? The role of rRNA is, which is called as ribosomal RNA, is to synthesize peptide bond. Now, if think about it, if amino acid one is coming by the tRNA, is brought by tRNA to the site of ribosome, then amino acid 3 is coming, sequence of amino acids, they are coming on the ribosome. So the question is, who will stitch the bond? Who will stitch the bond between amino acid 1 and amino acid 2? Who will stitch the bond between? And what is that bond? This bond is called as peptide bond. So two amino acids are connected by peptide bond, which is a covalent bond. This peptide bond is synthesized by rRNA. So rRNA, the function of rRNA is to generate peptide bonds in consecutive amino acid. So I just repeat, mRNA serves as a sequence of amino acid, tRNA serves as a carrier delivery person to bring amino acids to the ribosome, and on the ribosome there are already ribosomal RNA that tries to generate covalent bond between successive amino acids, and then they generate a particular protein. So let's take one example. So let's say there is a sequence of ribonucleotide in the RNA, AUG, GGG, AAA, UUU. So I'm trying to simplify this, and let's say CCC. So in 99% of the proteins, the starting amino acid is methionine. I'll come back to this. Starting amino acid is methionine. So now what in RNA codes for methionine? So in mRNA, AUG called as the triple nucleotides code for methionine. And methionine is generally the first amino acid in 99% of the proteins. Then the second amino acid is GGG. This codes for an amino acid glycine. Then AAA is responsible for coding for amino acid alanine. UU codes for phenyl PHE, which is codes for phenylalanine, and CCC codes for an amino acid called as proline. So what did I do here? This is a sequence of mRNA. This is a sequence of mRNA. AUG, triple G, triple A, triple U, triple C, and this triple nucleotides serve as a code or codon for amino acids. So you can imagine that there's a sequence of mRNA on the ribosome, and all these, this is a sequence of mRNA on a ribosome, and all these corresponding amino acids are brought on the ribosome by specific tRNA, and rest, the stitching of peptide bond is done by ribosomal RNA. So as I mentioned to you, that these three are, protein synthesis is a teamwork initiated by DNA, which is present in the in the nucleus in the case of eukaryotes, and the cytoplasm in the case of bacteria. Regardless, the DNA is transcribed undergo transcription by RNA polymerase to generate three kinds of RNA: mRNA, tRNA, and rRNA. mRNA serves as a template for amino acids. tRNA brings the specific amino acids, and ribosomal RNA develops or synthesizes or catalyzes the peptide bond between consecutive amino acids. So I mentioned to you that there is a code that triple nucleotides code for an amino acid. This code is called as genetic code. Genetic code. So this was discovered by Hargobind Khorana, who was at MIT and University of Wisconsin, Madison, and he developed this genetic code. What is genetic code? Genetic code delivers the relationship between mRNA and the corresponding amino acid. So Khorana got the Nobel Prize for discovering the genetic code, and this genetic code is conserved between viruses, bacteria, plants, and animals. So it's a big work. So I would like to draw some interesting point here. A virus could be DNA and RNA. Plant, a bacteria, plant, human cells, they're always DNA-based genetic material. But despite what is the genetic material, DNA or RNA, genetic code is very, very universal across the species. It is conserved. So in the next, I'm showing you how this relationship develops. And so relationship between DNA and proteins, and you know proteins are made up of amino acids. So this is very simple arithmetic. So DNA has 4 nucleotides, like A, T, G, C. Okay. RNA has 4 nucleotides too. A, U, G, C. So 4, it makes sense. The ratio is 1 to 1. A, T, U, G, G, and C, C. But the interesting thing is that there are 20 amino acids. So the question I'm asking you that how does these four bases, they make 20 amino acids? So there must be some rule. So 4 nucleotides of mRNA cannot make 20 amino acids, right? It cannot make 20 amino acids. So let's say if there is a combination of 4 times 4, then it becomes 16. 16 is also 4 less than 20 amino acids. So even this formula won't work. So what Khorana did, that he said that probably there are combination of three times combination, meaning 4 multiplied by 4 multiplied by 4, which is equivalent to 64. And in this 64 condition, these nucleotide will exist as triplets. Meaning that, as I showed you example, that triple A will code for alanine, triple G will code for glycine, so on and so forth. So now I would like to take you to the slides, lecture slides. This is chapter 2. The objective is explain how information in DNA is translated into protein that I discussed with you just now. You know what is transcription and what is translation, which enzymes are involved that I discussed with you. Compare and contrast mutagen versus mutations. Explain junk DNA. So we will talk about this here. So I switched on to these slides to highlight the table called as genetic code. This genetic code table, so you see here, there are triple nucleotides. and each triple nucleotides code for an amino acid. So there are 20 amino acids. So let's go to amino acids. I have shown you this earlier, but there are 20 amino acids and they differ in their chemical structure. So there are 20 amino acids. They have same NH2 group, same carboxylate group, and the 20 amino acids, they differ in their side chains. So all these highlighted with yellow shade, they are side chains, which is variable in the amino acids. So, so if we go back and forth with the slides, so if you see that in the genetic code, it is triple U or UUC that codes for PHE. PHE stands for an amino acid called as phenylalanine, and a phenylalanine, it looks something like this, right? So phenylalanine, it looks like this. So that's the relationship between the genetic code and an amino acid. And as I mentioned to you, so now one quick, very important point from exam point of view. When the DNA is replicating, the two strands of DNA give rise to two strands of new daughter DNA. But in transcription, it is only one. So instead of two strands, so DNA is double helical molecule. In this double helix, only one of the helices would serve for transcription, that you would say, and this is carried out by RNA polymerase. And there is a complementarity. Triple A gave rise to triple U, and then C would give rise to or give rise to an RNA molecule with G, T with A, and A with U. So this is a good diagram explaining that concept. So to summarize this chapter, which is from genes to protein, DNA, a blueprint of life. It is transcription and translation. And secondly, talking about mutations. Mutations are mostly bad for individuals. There are some good mutations also, but here I would just mention one very well-known mutation, which is called as sickle cell anemia, which occurs where due to a mutation in single nucleotides, and that nucleotide changes the amino acid, and the consequence is huge. What is the consequence? So hemoglobin, if it looks spherical, in case of mutation, it becomes sickle-shaped. So there's a dramatic consequences on the structure of hemoglobin. And this hemoglobin, which is sickle-shaped, cannot store oxygen. And hence, a person born with this mutation will have clinical problems. And there are some mutations, as I mentioned to you, which are very good. There's a mutation called as Milano mutation. This mutation was first found in a city of Milan in Italy. That's why it is called as Milano. And in this mutation, people having this mutation, they tend to have less deposit of cholesterol in their arteries. So they are less sensitive to, they are less prone to heart diseases, particularly cardiovascular diseases. So some mutations are good, some mutations are bad. Mostly mutations are bad. So with this, we finish chapter number 2.